Okay, welcome to our first talk of the ICG lab talk series that we have on a monthly basis. This is the first talk. Today we have it's a pleasure to introduce Bernhard Kainz. Bernhard Kainz is a lecturer in the Department of Computing at Imperial College in London. Bernhard's field of expertise is high-performance medical data analysis with real-time image processing techniques. Bernhard finished his PhD at Graz University of Technology. We actually did it at the same time, lab, and now Bernhard is in London. All right, so Bernhard, we're looking forward to your talk. Bernhard is going to talk about efficient motion compensation for fetal MRI.
Yeah, that's true, but first I think we have to check. Can you see my slides or can you see only me? Hello?
Yeah, could you say it again?
Yeah, can you see my slides or can you see only me?
Oh, we only see you. You need
okay,
to turn on.
So I share my slides. So can you see my slides now?
Yes.
Okay. And can you still see me?
No.
Okay. So you will get just the audio and not see all my nice gestures I do with my talk and so on. So I think you need to live with that. I'm sorry for that. So I'll try to do this yeah, in the most straightforward way. So this is a quite unusual setting for me because usually I don't wear headphones during talks and normal loudspeakers are quite out of fashion anymore. So the whole Department of Computing at Imperial has no standard PC loudspeakers, not a single one. So I have to wear these headphones. So as Mark already said, he asked me to give a talk about our ongoing research during the last few years on motion compensation and motion modeling for fetal MRI. This research is kind of the core for a lot of international research and worldwide ongoing research on the developing human and how, well, on the one side, how to improve diagnostics, and on the other side, of course, also how do we actually evolve from the very first step on, and maybe we find also reasons why certain diseases develop during the course of adulthood at some point. So, but in this talk, I will more talk about the core techniques we are using, so more from an algorithmical point of view, and what you can do with these. And if I have some time, I will show you maybe a few examples what people do with this at the moment. But the core will really be how to do motion modeling and these things. Okay, just a moment. So, did the slide forward now? So do you see another slide?
Yes.
Okay, good. So everything is working. So Mark, you already introduced me. Thank you very much for that. I'm currently lecturer here at the computing department, which is equivalent to assistant professor in the US. So basically, I'm heading my own group now here. And before this, I was basically a senior research fellow at King's. So I was working directly at the scanner, which is quite useful if you're working in medical image analysis, if you can steer the image acquisition directly. And before that, I moved actually to London with a Marie Curie fellowship after my postdoc time at Graz. And as Mark already said, we did our PhD together at Dieter's group in Graz from, well, until 2011, I think, approximately. Mark, another question. How much time do I have?
45 minutes plus questions.
45 minutes, okay. So let's dive into that. Why would we do something crazy like fetal MRI? Some of you might be already familiar with getting children. Usually what you do is 2 ultrasound scans at week 12 and week 20 of pregnancy. This is usually done by a sonographer. They are quite experienced. They can quite quickly get the images acquired which they need to assess if your baby is healthy or not. So why would we actually do something like fetal MRI? It's quite uncomfortable. You have to imagine that you need to get a pregnant woman between 20, well, pregnancy gestational age between 20 and 38 weeks into this small cavity here from an MRI scan. This is quite uncomfortable because, I mean, I don't know if anybody of you has been pregnant before, but if you have a belly like this, then it's not very comfortable to lay on your back. So we need actually to place them semi-laterally into the scanner so that we avoid pressure on the vena cava inferior and so on. So it's quite cumbersome. And during the scan itself, well, it takes an hour. So lying in there an hour is quite exhausting actually for pregnant women. An ultrasound scan would usually takes maybe 20 minutes, like here on this side. So the main problem we actually have with scanning fetuses in utero is that we have no control over the moving fetus. Usually, I mean, if you place an adult into the scanner, you just tell them, well, please don't move. If you move, you will corrupt the image and we need to do it again. So please don't move. Sometimes you even say, please don't breathe for 20 seconds or so. That's just impossible with a fetus. The fetus will not care and continue either doing this kind of party like this one. In the ideal case, they are sleeping and then it's fine. But this is already kind of a medium bad case where the fetus moves around a lot. We are always dealing with very, very small structures. So this would be approximately a fetal heart. In fact, this is a fetal heart of a pig, but a human fetal heart wouldn't look very different. So, I mean, I don't know how familiar you are with MRI, but MRI usually has a resolution around 1 to 2 to up to 4 millimeters per voxel on each side, and this structure is maybe a centimeter across. So this is already quite challenging for a modality like MRI. We have a challenging signal. If you have a moving object in your scanner, then you're not only introducing severe artifacts, you're actually also modifying the physical principles of these image modalities. So if you move the magnetic field around, what you get are actually intensity inhomogeneities. And this is something we also need to fight a lot to just get useful data out of the scanner directly. So we have a quite challenging signal overall for a sequence like this. And well, of course, also MRI is not as available as, for example, ultrasound. An ultrasound scanner is a small device like this. You can deploy it anywhere, like here, somewhere in Africa. No problem. It's well, kind of affordable. I wouldn't call it cheap, but 50K once is much better than 2 or 3 million once for purchasing an MRI and €1 million maintenance every year. So availability is also a big issue. And last but not least, of course, I mean, have you ever thought about doing experiments on pregnant women? It's already hard to do that on humans and adults. But on pregnancy with another human being inside her who has no kind of decision-making capabilities, ethics is a quite challenging topic for these kind of experiments. However, MRI has quite some distinct advantages over ultrasound. So first and foremost, we have really, really good tissue contrast, not only hard to soft tissue, but also within soft tissue, really, really nice contrast. That's also why it's usually used for adult brain imaging, because you can nicely separate the cortex from white matter and so on. You get a really nice and large field of view. In ultrasound, you usually see either the head or maybe some part of the torso or maybe one leg. So this is not the case in MRI. In MRI, we can see the whole fetus at once if necessary. And well, we have the option for comprehensive 3D imaging. This is the challenging part. So this is what we are actually working on. But there is the opportunity to do the whole fetus at once in a 3D volume, which has, of course, distinct advantages over single slices in ultrasound, or even 3D ultrasound, which has only just a small field of view. We can do functional imaging. Ultrasound is limited to flow. Doppler ultrasound, you might have seen that when you see in the umbilical cord the blood flowing, and in the heart the blood flowing, this red-blue separation where you can do in-plane measurements of how the blood flows. So we can do much more in MRI. We can not only measure flow, but also connections in the brain and even functional imaging. I mean, I wouldn't go that far and say we can look into the fetal brain, what he or she is thinking, but we can at least do something like a resting state fMRI and look how certain parts develop functionally. And we have various physical options to vary contrast. We are not limited to a single black and white scheme like it's done in ultrasound, but we can just adjust whatever we like to image and see different parts in different, in nicer representations. So to make one thing clear, we in our projects are generally not aiming to replace ultrasound for screening. Ultrasound for screening will be the non plus ultra for the next decades, very likely to be for the next decades because of its cheap price, availability, and easy application. So what we actually try to do is to improve this screening by learning how the human develops and how the fetus looks like at every stage of gestation. So with this knowledge, we can derive this knowledge from large populations of volunteers and also clinical cases. And with this knowledge, we can feed this knowledge back into standard screening procedures and compare actually to how it should look like in a healthy case, how it should look like in whatever disease case, whatever likelihood. So we use a lot of machine learning for this and also quite advanced image analysis. So just to give you a bit more context and a bit more motivation, so I'm sorry I only know numbers for the UK because one of our main funders is in the UK, and for them you always need to emphasize how important this is in the UK. But just to give you a context for what's actually targeted. So the target detection rate numbers for screening programs in the UK is that you detect at least 50% of all cardiac abnormalities, 60% of diaphragmatic hernias, so these are holes in your diaphragm, and 60% about lethal skeletal dysplasias. In fact, what's currently happening is that they achieve barely 35% of detection rate for major cardiac abnormalities, for example. So this is quite a big issue here because the even this kind of low target rates, I mean 50% is a chance 1 in 2 that you pick up a problem at an early stage, but even this is not achievable. But if we could screen all of them by fetal cardiologists, well, then we would likely hit this target, at least 1 of 2, but we have only 19 of those in the UK, and I'm pretty sure there are maybe only 4 in Austria. So it's a quite challenging topic. So we actually want to support and get all this knowledge into a standard screening procedure so that even you and I could do this screening based on this knowledge and make some quite informed decisions on the health status of this fetus. So as I said, we try to achieve this by atlasing. So this could mean that we could guide standard acquisition by knowledge from MRI. So let's say we have a nice MRI atlas which has all the labels which are required. In this case here, we depicted the spinal cord just for illustration reasons, and some ultrasound modality which can also give us some nicer field of view. This is also just for illustration. But if you have this information, we can simply guide an operator along whatever structure and tell them when diagnostically interesting and diagnostically important data has been acquired at a very early stage, so we can also make these kind of studies comparable. But I'm not here actually to talk about ultrasound. What I'm going to tell you is how we actually take fetal MRI, because I mean, if you would put a lot of brain power into standard MRI sequence development, and this is actually currently also done, then you will likely come up with sequences comparable to other sequences. So if you just try a naive approach and do a stack of slices, for example, one after each other, and try to acquire a volume of the moving fetus, then with quite some tweaks, you might be successful with these individual slices. So the individual slices might look quite well. So there is only little motion corruption. You have a kind of snapshot. So these are just 3 consecutive slices of one of these stacks. and they look quite okay. But the problem is, MRI is quite limited in with real-time capabilities. So each of these slices, if you do it right, will take somewhere between 50 and 300 milliseconds to acquire. It depends a bit on the field of view and whatever other conditions you have. So what's likely that between the acquisition of the individual slices, when 300 milliseconds pass, the fetus is very likely to have moved. So what happens then is, well, within the native slice orientation, you might still see nice structure, but as soon as you try something like multiplanar reconstruction or just want to look from a different perspective in at your fetus with a differently oriented cutting plane, then your slices will very much look like this. So this is these are our motion artifacts. The kind of more regular streaky ones are caused by maternal breathing, and this is anyway a nicer case, so we can still somehow see that there is a brain, but in any case this is diagnostically not useful. So here we can't really see anything or make any conclusions. So if we would look at this in 3D, I mean, it would look awful. So we don't have really any idea. Not only that I was lazy and didn't define a very well-defined transfer function. This is MRI, so it's even trickier. It's just hard to see any of the structures you're interested in from this brain, for example. So conventionally in MRI, so these slides partly originate from a talk I gave recently for MRI physicists. Forgive me that there are some MRI physics expressions in there, but there are options to do motion compensation in MRI. So usually you would use this for compensation for breathing motion or involuntary movements. The heart, for example, you can't stop, so you need some other options, or some other patients just can't hold still, or very small children have also problems with it. So either you can place either a navigator, which means you put a small scanning area somewhere, usually in the diaphragm, and track certain intensity, which lets you conclude on the actual motion of the lung, for example. But in our case, the offsets are just too far. Your respiratory motion is usually not more than 1 centimeter or 2 in offset. And we are dealing with more than 2 or 3 centimeters offset and really hard motion, so that's not doable. Gating is also something which is used, for example, for cardiac imaging, where you just take a single image or a stack of images at a single point of, for example, the cardiac cycle, so you know more or less how the shape of your heart, for example, is at a certain point, and then you acquire the slice, and then you reassemble after a longer scan time, after many cycles, your actual volume at a certain state. It's not possible, because how do you want to gate, how do you want to track fetal motion? So we can't just, for example, put on external sensors, some reflective markers on the fetus. That's just not possible. Motion robust 3D sequences are developed, so these are sequences which recover for small offsets, like, for example, Parkinson patients would have this problem with holding still when their brain is scanned. So there, for this small kind of random offsets, you can develop sequences which are motion robust in Fourier space. This is where actually MRI acquires their data from. Still, we have here the problem of bulk motion. So the fetus usually moves a whole part at once, and not just a small tissue area. So case-based assumptions just don't hold here. And fast sequences, well, as I mentioned before, if you do something with a lot of tricks for sequence development, then you can come up with images which are nice in 2D, but we have, again, the problem of motion between the slices. And this is actually exactly what we use. So we use fast sequences to at least acquire nice 2D slices of the moving fetus, and acquire kind of one slice at a time to freeze motion, at least within this 2D image. So currently we are mainly working on T2 weighted imaging, because the brain is currently one of the kind of most interesting areas, and T2 just gives you a very good contrast for the brain. But we are also working on thorax and abdomen, which works also quite well in T2. We use a sequence which is called single-shot fast spinecho, which is approximately 100 milliseconds. By now, we started around 500 milliseconds. Now we're at 100 milliseconds by slice, but we still have slices, the motion artifacts between slices. And well, in reality, there are, of course, also things like we scan action packages, use sense compression and parallel imaging. So we acquire several images at a time, but far apart from each other, so that all the physical assumptions are still hold true, and that we don't actually affect our slices by exciting certain areas. And then we do a certain trick. We just oversample the input space wildly and hope that at least by acquiring a lot of these slices in orthogonal directions, in any directions we can do, that we sample at least every part of the fetus. Even if the spatial coordinates, if the spatial location of this information might not be correct yet, we hope at least by doing this to sample all of the relevant parts of the fetus. So let's say we have now a few of these stacks. So these stacks of slices usually consist of parallel slices and motion corrupted data. So we have, let's say, 4 orthogonal slices here, and what we actually try to achieve is to realign all of the slices with respect to one of these slices as a template. So ideally, we try to calculate and optimize the motion out by having a heavily oversampled input space. Our general pipeline is, actually, it is quite simple. These are all, these kind of approaches are all called slice to volume registration approaches. You can use them for various kinds of motion compensation if you have exactly this kind of problem. But what we do, in fact, is, well, first of all, I'll come back to this later. First of all, we need to select a specific region like the brain. So we currently, with this approach, we can only reconstruct for one bulk of motion. So something like the brain and the head is a rigidly enclosed compartment, and the fetus moves it at once. So you actually have to select first one of these regions to fulfill a few assumptions which are required later. But despite this, let's say we have a generous identification of the brain. We start first by just aligning the stacks. So what's also likely is that the fetus might have moved between the acquisition of individual stacks, but this is kind of recoverable, because if the fetus just as a whole moved a few centimeters in each direction, we can recover this usually by just using volumetric registration. And we only need to kind of recover for a whole bulk, and we don't need to be very accurate at this time. But then we start the magic. So what we do is we first try a partially reconstructed volume by apply techniques like super resolution and co-aligned slice alignment with point split functions, which gives us, I wouldn't call it average, but something like an initial guess of how our structure actually could look like. It doesn't look like this. This is already a very late stage of the reconstruction. It looks more like a very, very blurry and corrupted version of that. But it's usually good enough to at least figure out where brighter, darker areas are, so that you can start some kind of registration and realignment of the slices. So what we do at this stage, we break the parallel alignment of the slices within the stacks, and we realign each of the slices individually to fit this initial guess of our volume a little bit better. We are not aiming for perfectionism at these stages. We just try to make it a little bit better to fulfill or to kind of model the motion here. So slice to volume registration is basically quite simple. It's an intensity-based approach. You can implement it as however you like. Usually we use something like a gradient-based cross-correlation attempt. But this is just a detail. The problem with slice to volume registration is that non-rigid deformations are quite ill-defined. So if you think about that, if you move a volume within a slice or vice versa, a slice in a volume, in which direction would you bend the slice to fulfill certain conditions? There are some approaches, and I will also show you later one, which is more robust towards elastic motion, but for this step, we just assume rigid slice to volume registration. So and then we improve this a little bit. What you can do next is just to go back to reconstruct again using a measured point split function, and then to get a model which is a little bit better. So if you look at this, there is the very first stage, very blurry, ugly version of that, and then after time, after a few iterations, it starts to improve and fit quite well the actually measured data. So this, actually the final scene probably never happened like this, right? So this is just the optimum how all the acquired slices in orthogonal directions fit best to each other. The fetus might never have looked like that. Well, the structure will have looked like this, but the fetus might never have gazed in this specific direction. So the general problem, how to solve this, is basically an inverse problem. So ideally, we would be able to invert a kind of acquisition protocol like this, or an acquisition model like this, where each slice is defined by a sampled volume, specifically sampled volume. So we are not just linearly interpolating, but an MRI has quite a specific point split function, which is like a filter kernel, but actually orientation dependent. We currently use a sink in plane and Gauss through plane, but basically this is a looks simple equation, which would be nice if we could just invert it, but it's simply not possible. So what we need to do is to solve this kind of inverse problem and to find basically a minimum between slices we can simulate by just looking at the similar orientation of the slice in our temporary volume compared to the actually acquired slices. We are taking here the L2 norm, but you could take any norm here. It's just mainly the difference. But the problem with such kind of iterative optimization problems is, of course, if you don't do this carefully, you would end up amplifying a lot of noise as well, and also sometimes maybe the registration might not work that well. So you would amplify quite a lot of noise. This is, of course, also why you need some regularization. We currently, there's really the most simple implementation we have currently running. We use a quite basic age-preserving filtering for this regularization, which is based or quite similar to anisotropic diffusion. This helps you to handle noise and to do the optimization and this solving of this inverse problem a bit more accurately. So this pipeline looks, just to illustrate this, the iterative optimization a bit more here. So if we just generate a volume which is blurry and just based on the point spread functions and motion corrupted, like the one you saw before, or this one, for example, then we can just initialize our optimization volume with that. And if we just sample out one slice, which is correspondent to the orientation and location of the slice we actually acquired, and we can just compare them and we'll get some difference image. And each of these differences can be fed back into this volume by using an inverse version of our point spread function and update each of the boxes accordingly, so that most voxels will start to agree at some point on a specific location, on a specific intensity. However, the problem is still, the registration is not perfect strategy. Well, it's not possible to implement it in a perfect way. So what it's actually doing is just a very convex optimization, and a simple gradient descent might also sometimes mislead slices, and some slices might just not be possible to be registered, because, of course, I didn't tell you the whole truth about 2D image acquisition. We can do it for almost all of the slices without motion artifacts, but if the fetus moves really, really, really fast, we still suffer signal loss, and you will get black areas in your 2D slice, and so areas which you simply can't register, and they will produce artifacts. And to cope with these artifacts, automatically we usually use some robust statistics based on aim estimators. I'll show you later what I mean by that. But basically we train several multi-layer model on top of our super resolution reconstruction to get some idea, some unsupervised idea, how true the currently derived or reconstructed value is, and if we can trust it or not. And with that we get weights so that we can upweight or downweight parts. So if you would just use it without robust statistics, you would get all this kind of streaky artifacts which are misregistered slices, signalers, and so on. But if you do it with outlier rejection, with automatic outlier rejection, you will lose quite some data, but that's why we do a highly redundant data sampling approach, and if we have enough data, we can afford to throw away 30–40% of our required slices. So if you do that without it and just leave the very reliable ones, our results are actually quite good. So robust statistics is, as I said, based on M estimators. We basically train an EM model on various levels, so on a voxel level, which is based on the differences to the actually required data, on slice level, so we can throw away whole slices or just downweight for whole slices. So our model is actually the blue one. There were other models which just did weighting. We, at a certain point, so this, the take-home message of this is just at a certain point we weight pixels towards the likelihood that they are representing the true measured data, and at a certain point when they are getting too different, then we just completely reject them. Yeah, other models are just weighting them and try to downweight them in a certain way. So let's have a look here. So we are okay. So I'm running out of time. We also have to take care about some bias correction things. I will not go into detail. That the problem can also be that sometimes some intensities might be differently distributed in some slices than others. So we constantly run bias correction similar to N3 and 4 bias correction, which is quite popular for especially high field brain imaging. 7 Tesla, for example, always needs bias correction simply because you can't achieve the field inhomogeneities. The field homogeneity which would be required to do proper image acquisition. So since this is a nice approach and quite independent, so all the slices are quite independent from each other, it's very easy to parallelize this. We basically use standard approach, kernel level parallelization procedures, which parallelize this per slice, so we can do this quite efficiently. This is especially important if you acquire quite a lot of data. It's getting infeasible at some point to do all this registration, all the time, this cost function optimization even on CPU clusters. So we just do it on multiple GPUs, and it achieves quite good performance. So we are not yet at real time, but we can do these reconstructions within a few minutes. As I said, we don't use just simple interpolation kernels. We have point spread functions. These are actually measured from the scanner, so they are quite comparable across scanners, but if you do it specifically for your scanner, then it's the results improve a little bit structurally and the edges improve a little bit, but the general idea is you don't use a simple linear interpolation kernel. So, and as I said, we are currently restricted to just rigid subregions of the area of the fetus which you can reconstruct. And this is quite some limitation because you need to do some manual registration, which is cumbersome, lots of work, expensive, and so on. So that's why we recently developed another method. Since we saw that we have quite some computational resources left on our GPUs, we just increased the redundancy even more, significantly more, and just split the whole image into small, very small, but overlapping patches, for example, this kind of square patches, and did a similar approach to slice to volume registration on these patches, but with an additional layer of robust statistics to get rid of very redundant patches. And the patches are actually carved at some point to get rid of information which gets misregistered at some point. And this is a naive approach to do whole uterus reconstructions. A more intelligent approach would be if you would already use semantically meaningful information, and there is quite a lot in computer vision literature. So something like superpixels are quite good representatives of areas which seem to belong together. In fact, it's nothing else than a multi-level level set, but turns out that these kind of techniques work quite well to, first of all, reduce our redundancy a little bit again, and to get already structures which are very likely to contain consistent information. I mean, this is, of course, a showcase example. So if you look at this superpixel, it contains already the whole brain and nicely fits to the skull, and can be used directly to reconstruct the brain. But even if this would split the brain into two, it wouldn't matter, because they would also just use move consistently through the registration. And this is also something we can actually use to estimate areas of bulk motion. So areas which are rigidly connected, like the brain, or partly rigid, maybe only elastically or thinly connected, like the thorax or the lower abdomen, by just tracking the statistics on the patches. So the less well they are able to be registered, the more likely it is that they are in some area which just contains crap, yeah? So like amniotic fluid or the maternal tissue around. So the less likely it is to contain something useful. So if we keep track of the probabilities from our M estimator, we actually can already derive a rough estimate for which areas would be relevant for bulk motion, and then we could use this, of course, to do classical slice volume registration or refinement to get more accurate, more context for the registration, so more structurally accurate reconstruction. And well, this approach is quite applicable. So as you can imagine, it's even harder to do it for twins. So these are twins in the uterus, and if we just reconstruct the whole uterus, we have everything we are interested in at once. So what we can see here is the fetal brain, and there is another fetal brain from the other fetus. This is the lung from the other fetus. This is the placenta around here. And if you look closely, the first fetus here has some problem with the kidney. So this is something which was completely new, because it was very hard to this point to reconstruct structures like the kidney. If you remember the heart from before, the centimeter structure, a kidney at this age, so these two are around 28 weeks. So a kidney at this age is not even half a centimeter on the long side. The short side is maybe a few millimeters. And this is a cystic kidney, which was very hard actually to investigate before these kind of techniques. So I have 2 minutes left. I will spare you the boring data. So of course this stuff breaks down at some point. So this will break down around 4 centimeters motion amplitude. So if the fetus moves more than 4 centimeters consistently over the whole sequence, we cannot reconstruct, but before that we are actually quite good. We are reconstructing quite well. We are doing these kind of experiments on synthetic motion. So we actually take a data set from an adult brain, for example, and artificially motion corrupt it, and then try to reconstruct so that we have a ground truth, because otherwise from fetal data it's quite hard to get ground truth. The patch-based approach is also quite good if you look at Femur Transformation. So this orange one is the old one, and these two are variations of the patch-based approach of the new one. So we gain at least 20% in accuracy for longer time. So this breaks down much earlier than the patch-based approach when it comes to this kind of a thin motion. I mean, it's a pity that you can't see me, because I usually do these gestures during my talk and show you how the fetus moves, but just imagine how I move my torso now. So there is some kind of a thin elastic motion involved, which you couldn't just reconstruct by using standard slice to volume reconstruction. So this is currently used quite widely. We have big projects which all rely on that. For example, studying the human connectome at an early stage, so how develop all the connections in the human brain from 20 weeks or even 18 weeks now up to 2 years, 3 years, and then through adulthood. We are doing whole body MRI, trying to improve ultrasound imaging by atlas building from MRI. This is what we're doing in this project, that we are currently looking at all other organs as well, lungs and major cardiac vessels like these here, like the spinal cord, liver and kidneys are interesting. The placenta is also interesting with the page-based approach because this is something where standard slice-based approaches fail completely. Placenta is an organ which has just no defined shape. It's not similar from person to person. So this is also quite challenging here. So if you want to try out the code, it's on GitHub. We will soon update it with another branch of page-based reconstruction. So it might be maybe also applicable to other programs which suffer motion corruption. So in summary, well, it works currently quite well. We have it distributed all over the world. People are quite happy with what they can do at the moment. Bioengineers are happy with tools you give them, and they just throw in their data and something useful and nice comes out by just a small command line. But it's a command line tool, so it's not very, very fancy. So do you remember this one? Yeah. Very corrupted data. After reconstruction, we have a nice fetus. Yeah, and if we do whole uterus reconstruction, we even see the whole baby. So a few references. And with that, I say thank you very much for your attention. Thank you. So yeah. Let me go back to this. So here, now I can see you. Upside down, but okay. Okay.
Okay, well, thank you very much for this really interesting talk, Bernhard. We're open for questions, or Bernhard is open for questions.
Yeah, give me a moment. Maybe I can.
Yes, we see you now again.
Okay, good. So this is what I meant. I do this. Yeah, I paint my torso, and this is how the fetus moves.
Okay, questions. Then I will start with the first question. So everything you do is done on the GPU, so it's highly. Can you hear me? It's highly parallelized.
Yeah.
But is performance still an issue? So performance in terms of time, not accuracy?
So the performance is definitely an issue. Currently, we have a post-processing pipeline, yeah. But in the long run, we actually want to integrate these kind of approaches into the scan process directly. I mean, time is money, yeah. So if you do one of these scans, just maybe focusing on the brain, it takes you 10 minutes scan time, and our scan slots are 60 minutes. If you could integrate these approaches directly in real time into your scanner, then you could actually tell the scanner where you still miss data, where you would need to acquire more data. So you could achieve something like a differential data acquisition pipeline, which would be, of course, much faster and also more accurate. Because, I mean, the presentation is nice, you see it works somehow, but what I didn't tell you is it works in 80% of the cases. In the other cases, it doesn't, because we just miss data. We do blind data acquisition and hope that we acquire all the data. Yeah? But if we could kind of guarantee that this now worked, that we have all the data we need to reconstruct while we reconstruct, yeah, we can do an early stage reconstruction, for example, during scan, then we would need real-time or close to real-time performance. So this performance would be very, very much desired for this kind of attempts. It would actually pave the way for completely new scan sequences. So it's quite important. And also the latter approach I showed you, this patch-based one. So it's kind of quadrupling your algorithmic complexity. So if you run the initial approach, lies the volume on maybe, let's say, 16 CPU cores, it takes you 2 or 3 hours to do the reconstruction. On the GPU, just for the brain, it takes you 3 minutes. And if you would do this patch-based approach, it would take you 2.5 weeks to do one of these uterus reconstructions. This I can do on the GPU, just maybe it takes now 6 minutes or so. Oh, thank you. Oh, very nice. So parallelization is quite an issue here, and it's actually essential for these more advanced approaches to do the whole uterus, because 2.5 weeks is just not doable anymore.
Yeah. Okay, further question? Sorry? Yeah. Bernhard, you wanted to say something?
No? Okay.
More questions? Yeah, Alexander?
Did you are machine learning techniques an option? Because I think that it's a bit hard to get ground truth data for supervised learning approaches. So
we are currently exploring several strains of machine learning here. So this already, what I showed you, this fundamental basic technique here already employs unsupervised machine learning. So this robust statistics stuff, this is all unsupervised learning. It's an M estimator, so it's machine learning. What you might be referring to is supervised learning for approaches, for example, like super resolution, so very, very en vogue at the moment, deep learning for super resolution. And in fact, I just started a project about exploring this and to learn actually how structure looks like from the actually acquired data. So we are also using this, but then you have the challenge that you run the kind of reconstruction, the registration, some super resolution deep learning, and some deep learning inference, all on the same GPU. So this gives you even a bit more headache now to schedule all these things. So it's getting more complicated. But yes, we are considering lots of machine learning, and your second part was about the ground truth. So the ground truth here, our ground truths are the 2D image slices. We know everything we scan here, or almost everything, is the true anatomy. Yeah, so we can learn from that. 3D is hard, so there we can either use the successfully reconstructed brains or whatever fetuses and learn from there, or what we also currently explore is transfer learning back down from the adult back down into the fetus, so that we use trained models from there and go back into the developmental structures of the fetus.
Mm-hmm. Okay, further questions? Okay, I have one last one, which is, so we talked about the performance in terms of time, but how about the performance in terms of accuracy? How much room for improvement is there? Of course, there's an upper limit. If there is no more signal in the data, then you can't further improve it. But how big can the improvements be? What do you think?
So currently we are reconstructing with a performance of around 20 to 30 decibel in PSNR. PSNR is not the ideal thing to compare, because it just uses the global intensity information. We are close to 1, so around 0.9 for this, for example, structural similarity index, which is quite nice. We usually compare here again to the measured data. So there is one thing maybe which might be interesting in this context, because you said what's the upper bound here?
Yeah.
So from the sampling, from the structural information, the upper bound would be the data you acquire from the scanner, but that's not completely true. So we have some kind of incomplete Nyquist oversampling. Yeah? So we can't guarantee that we always fulfill Nyquist, but what you can do if you scan that heavily in all kind of directions is you can reconstruct on a much lower resolution and much more detail as you actually acquired the data. So if you acquire slices with I think we have currently 1¼ centimeters pixel size in each slice with 2 millimeters, sorry, did I see centimeters? 1.25 millimeters pixel size in plane and 2 to 2.5 millimeters through plane voxel size. So we can reconstruct usually, and this is what we actually also do, down to 0.5 millimeters isotropic voxel size. But then at some point you hit again Nyquist because it doesn't improve. So, but finding this point is a completely well, I would call it esoteric approach. Is something people who currently use these tools break themselves if they can hit this sweet spot immediately by just guessing it. So a good guess would always be something like 0.75 millimeter reconstructions, and this then actually gives you a better resolution as you would gain from the original slices because you do this heavy oversampling. But then it's questionable if it would improve going down 0.5, 0.45. So this is something you have to guess because you can't measure Nyquist at this point.
Sure.
But this is completely done empirically. But we kind of improve even the original scan sequence by doing this.
Mm-hmm.
Yeah.
Okay. Thank you. Okay, if there are no further questions, then let's thank our speaker again. Thank you, Bernhard. It was really a pleasure having you. Have a nice day.
Yeah,
I just need to close the session here. So our next speaker is next month on the 8th of November, Professor Jörn Kohlhammer from Fraunhofer ICG, and he's leading the visual analytics group there. and he will be here in person. Well, if you haven't signed yet, if you're part of the seminar, then please sign in the list. And yeah, we will see each other next month again. Thank you, Bernhard. Bye.